All right. Hi, everyone. I'm Kim McCall, and I'm looking forward to telling you about our work and cell death in the ovary. And we use Drosophila as a model system. So Drosophila has been around for about 100 years, more than 100 years, as a great model system. Some of its advantages are that it has a fast generation time, tons of genetic tools, and evolutionary conservation of human genes, in particular, 75% conservation of human disease genes. So it makes a great model for studying cell biology, developmental biology, neurobiology, gene regulation, et cetera. So in our work, in our lab, we study cell death, and cell death occurs throughout development of the fly and in response to environmental cues. And in particular, we focus on the ovary. And the ovary is a huge tissue in the fly. It fills up most of the female abdomen. They are egg machines. That's what they do. So this is what the structure of the ovary looks like. It's like an assembly line with the mature eggs all the way on my side here. A fly makes about 50 of those a day and lays them, so constantly producing eggs. And cell death is a normal occurrence in the ovary. So cell death occurs in many different cell types in the ovary, but we focus on two forms of cell death that happen in the ovary. One in response to starvation. This form of cell death can also be induced by drug exposure and other environmental cues. And this happens in mid-oogenesis. So right there where you see the 8, cell death can occur in response to these cues, and it undergoes apoptosis, which is a very common cell death pathway. This is very similar to follicular atresia in mammals. So that's the most common outcome for an oocyte in mammals. Later on, as development proceeds, there's a subset of cells, the ones with the big black nuclei there, that will undergo a developmental program cell death. So these cells always die for every egg that is made. And this cell death is non-apoptotic. It was very mysterious for a number of years, and we've spent a lot of time trying to understand how this cell death occurs. So these are some of our microscopy images showing you what this cell death looks like. So the one with the purple cells and the green cells are the germline, and the purple cells are somatic cells around the outside of the egg, the developing oocyte. And when death begins, you can see that the purple cells enlarge dramatically. They are engulfing the dying germline. And this is very similar to what happens with granulosa cells in follicular atresia in mammals. So the oocyte dies and granulosa cells engulf the dying oocyte. So on the side closer to me, that's the developmental cell death that happens. And what we found is that these dying germ cells die by acidification. So that red stain you see is a marker for pH, and they're becoming acidified, and they're actually being killed by the surrounding green cells. So very interesting form of cell death. And recently, Alan Spradling's group has shown that a very similar cell death happens in the fetal mammalian ovary, where germ cyst cells will die, leaving only the primordial oocytes. And so in the last 30 seconds, I want to tell you about a new area we've been working on to develop the fly as a model for reproductive disturbances after traumatic injury. So women have been found to have reproductive problems such as increased miscarriages and fertility issues. And we have found that by inducing traumatic injury in the fly, we see defects such as decreased ovulation and increased cell death. And so what we're hoping is that some of these pathways will be conserved in humans.